Cell, the unit of life. When you look around, you see both living and non-living things. You must have wondered and asked yourself, what is it that makes an organism living? What is it that an inanimate thing does not have? What a living thing has? The answer to this is the presence of the basic unit of life, the cell in all living organisms. All organisms are composed of cells. Some are composed of a single cell and are called unicellular organisms, while others, like us, composed of many cells, are called multicellular organisms. What is a cell? Unicellular organisms are capable of independent existence and performing the essential functions of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, cell is the fundamental structural and functional unit of all living organisms. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. Cell theory. In 1838, Matthias Sheldon, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissues of the plant. At about the same time, Theodore Schwann, in 1839, who is a British zoologist, studied different types of animal cells and reported that cells had a thin outer layer, which is today known as the plasma membrane. He also concluded, based on his studies on plant tissues, that the presence of cell wall is a unique character of the plant cells. On the basis of this, Schwann proposed the hypothesis that the bodies of animal and plants are composed of cells and products of cells. Sheldon and Schwann together formulated the cell theory. This theory, however, did not explain as to how new cells are formed. Rudolf Virchow in 1855 first explained that cells divided and new cells are formed from the pre-existing cells, omnis cellula e cellula. He modified the hypothesis of Sheldon and Schwann to give the cell theory a final shape. Cell theory, as understood today, is: all living organisms are composed of cells and products of cells. All cells arise from pre-existing cells. An overview of cell. You have earlier observed cells in an onion peel and or human cheek cells under the microscope. Let us recollect their structures. The onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cells of the human cheek have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane-bound structure called the nucleus. This nucleus contains the chromosomes, which in turn contains the genetic material, DNA. Cells that have membrane-bound nuclei are called eukaryotic, whereas cells that lack a membrane-bound nucleus are called prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called the cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both the plant and animal cells. Various chemical reactions occur in it to keep the cell in the living state. Besides the nucleus, the eukaryotic cells have other membrane-bound distinct structures called organelles like the endoplasmic reticulum, the Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. Ribosomes are the non-membrane-bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles, which are chloroplast in cells and mitochondria, and on rough endoplasmic reticulum. Animal cells contain another non-membrane-bound organelle called the centrosome, which helps in cell division. Cells differ greatly in size, shape, and activities. For example, microplasmas, the smallest cell, are only 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. The largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7.0 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular. The shape of the cell may vary with the function they perform. Prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO, which is pleuropneumonic-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cells. They may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, which is rod-like, coccus, which is spherical, vibrio, comma-like, and spirillum, which is spiral. The organization of the prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shapes and functions. All prokaryotes have a cell wall surrounding the cell membrane except in mycoplasm. The fluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition to the genomic DNA, which is a single chromosome, and circular DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called the plasmids. The plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistance to antibodies. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryotic cell, except for ribosomes. Eukaryotes have something unique in the form of inclusions. A specialized differentiated form of cell membrane called mesosome is the characteristic of prokaryotes. They are essentially enfoldings of cell membrane. Cell envelope and its modification. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure, that is, the outermost glycocalyx followed by the cell wall and then the plasma membrane. Although each layer of the envelope performs distinct function, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of the differences in the cell envelopes and the manner in which they respond to the staining procedure developed by Gram. Those that take up the Gram stain are called Gram-positive, and the others that do not are called Gram-negative bacteria. Glycocalyx differs in composition and thickness among different bacteria. It could be a loose sheath called slime layer in some, while in others it may be thick and tough called the capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membranous structure is called the mesosome, which is formed by the extensions of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and laminae. They help in cell wall formation, DNA replication, and distribution of daughter cells. It also helps in respiration, secretion processes to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes, like the cyanobacteria, there are other membranous extensions into the cytoplasm called the chromatophores, which contain pigments. Bacterial cells may be motile or non-motile. If motile, they have a thin filamentous extensions from their cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides flagella, pili and fimbriae are also surface structures of the bacteria but do not help in role in motility. The pili are elongated tubular structures made of a special protein. The fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rocks in streams and also to the host tissue. Ribosomes and inclusion bodies. The prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made of two subunits, 50S and 30S units, which when present together form 70S prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysomes. The ribosomes of a polysome translate the mRNA into proteins. Inclusion bodies. Reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie free in the cytoplasm. Example, phosphate granules, cyanophycine granules, and glycogen granules. Gas vacuoles are found in blue, green, and purple and green photosynthetic bacteria. Eukaryotic cells. The eukaryotes include all the protist, plants, animals, and fungi. In eukaryotic cells, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possess an organized nucleus with nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structures. Their genetic material is organized in chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as the former possesses cell walls, plastids, and a large central vacuole which are absent in animal cells. On the other hand, animal cells have centrioles which are absent in almost all plant cells. Let us now look at individual cell organelles to understand their structure and functions. Cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope in the 1950s. Meanwhile, chemical studies on the cell membrane, especially in human red blood cells, enabled the scientist to deduce the possible structure of plasma membrane. These studies showed that the cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar heads towards the outer side and the hydrophobic tails towards the inner part. This ensures that the non-polar tail of saturated hydrocarbons is protected from the aqueous environment. In addition to phospholipids, membrane also contain cholesterol. Later, biochemical investigations clearly revealed that the cell walls also possess protein and carbohydrate. The ratio of protein and lipid varies considerably in different cell types. In human beings, the membrane of erythrocytes has approximately 52% protein and 40% lipids. Depending on the ease of extraction, membrane proteins can be classified as integral and peripheral. Peripheral proteins lie on the surface of membrane while the integral proteins are partially or totally buried in the membrane. An improved model of the structure of cell membrane was proposed by Singer and Nicholson in 1972, widely accepted as fluid mosaic model. According to this, the quasi-fluid nature of lipid enables lateral movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the point of view of functions like cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, etc. One of the most important functions of the plasma membrane is the transport of the molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called passive transport. Nutrasolutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is, from higher concentration to lower. Water may also move across this membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. As the polar molecules cannot pass through the nonpolar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient, that is, from lower to higher concentration. Such a transport is an energy-dependent process in which ATP is utilized and is called active transport. Example, sodium-potassium pump. Cell wall. As you may recall, a non-living rigid structure called the cell wall forms an outer covering of the plasma membrane of fungi and cells. Cell wall not only gives shape to the cell and protects the cell from mechanical damage and infection, it also helps in cell-to-cell interaction and provides barrier to undesirable macromolecules. Algae have cell walls made of cellulose, galactans, mannans, and minerals like calcium carbonate, while in other plants it consists of cellulose, hemicellulose, pectins, and proteins. The cell wall of a young plant cell, the primary wall, is capable of growth, which gradually diminishes as the cell matures, and the secondary wall is formed on the inner towards the membrane side of the cell. The middle lamina is a layer mainly of calcium pectate, which holds or glues the different neighboring cells together. The cell wall and middle laminae may be traversed by plasmodesmata, which connects the cytoplasm of neighboring cells. Endomembrane systems. While each of the membranous organelles is distinct in terms of its structure and function, many of these are considered together as an endomembrane system because their functions are coordinated. The endomembrane system includes endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. Since the function of mitochondria, chloroplast, and peroxisomes are not coordinated with the above compartments, they are not considered as part of the membrane system. 1. The endoplasmic reticulum. Electron microscopic studies of eukaryotic cells reveal the presence of a network or reticulum of tiny tubular structures scattered in the cytoplasm that is called the endoplasmic reticulum. Hence, ER divides the intercellular space into two distinct compartments, that is the luminal, which is the inside the ER, and extraluminal, which is the cytoplasm compartments. The ER often shows ribosomes attached to the outer surface. The endoplasmic reticulum bearing ribosomes on the surface is called the rough endoplasmic reticulum, RER. In the absence of ribosomes, they appear smooth and are called smooth endoplasmic reticulum, SER. RER is frequently observed in the cell activity involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is the major site for the synthesis of lipids. In animal cells, lipid-like steroidal hormones are synthesized in SER. 2. Golgi apparatus. Camillo Golgi in 1898 first observed densely stained reticular structure near the nucleus. These were later named Golgi bodies after him. They consist of many flat, disc-shaped sacs or cisternae 0.5 micrometer to 1.0 micrometer in diameter. These are stacked parallel to each other. Varied numbers of cisternae are present in a Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distinct convex cis or the forming face and concave trans or the maturing face. The cis and the trans faces of the organelles are entirely different but interconnected. The Golgi apparatus principally performs the functions of packaging material to be delivered either to the intracellular targets or secreted outside the cell. Materials to be packed in the forms of vesicles from the ER fuse with the cis face of the Golgi apparatus and move towards the maturing phase. This explains why the Golgi apparatus remains in close association with the endoplasmic reticulum. A number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from the trans phase. Golgi apparatus is the important site for formation of glycoproteins and glycolipids. 3. Lysosomes. These are membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes, example hydrolases, lipases, proteases, carbohydrases, optimally active at the acidic pH. These enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. 4. Vacuoles. The vacuole is the membrane-bound space found in the cytoplasm. It contains water, sap, excretory product, and other materials not useful for the cell. The vacuole is bound by a single membrane called the tonoplast. In the plant cells, the vacuoles can occupy up to 90% of the volume of the cell. In plants, the tonoplast facilitates the transport of number of ions and other materials against concentration gradient into the vacuole. Hence, the concentration is significantly higher in vacuole than that in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In many cells, as in protist, food vacuoles are formed by engulfing the food particles. Mitochondria. Mitochondria, unless specifically stained, are not easily visible under the microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cells. In terms of shape and size also, considerable degree of variability is observed. Typically, it is sausage-shaped or cylindrical, having a diameter of 0.2 to 1.0 micrometer, average 0.5 micrometer, and length 1.0 to 4.1 micrometer. Each mitochondria is a double membrane-bound structure with the outer membrane and the inner membrane dividing its lumens distinctly into two aqueous compartments, that is, the outer compartment and the inner compartment. The inner compartment is filled with a dense homogeneous substance called the matrix. The outer membrane forms the continuous limiting boundary of the organelles. The inner membrane forms a number of infoldings called cristae towards the matrix. The cristae increase the surface area. The two membranes have their own specific enzymes associated with mitochondrial function. Mitochondria are the site of aerobic respiration. They produce cellular energy in the form of ATP, hence they are called powerhouses of the cell. The mitochondria also possesses single circular DNA molecule, a few RNA molecules, ribosomes of 70S, and the components required for the synthesis of proteins. The mitochondria divide by fission. Plastids. Plastids are found in all plant cells and in euglenoids. These are easily observed under the microscope as they are large. They bear some specific pigments, thus imparting specific colors to the plants. Based on the type of pigments, plastids can be classified into chloroplast, chromoplast, and leucoplast. The plastids containing chlorophyll and carotenoid pigments, which are responsible for trapping light energy essential for photosynthesis. In the chromoplast, fat-soluble carotenoid pigments like carotene, xanthophylls, and others are present. This gives the part of the plant a yellow, orange, or red color. The leucoplast are the colorless plastids of varied shapes and sizes with stored nutrients, such as amyloplast store carbohydrates, which is starch, example potato, store oils and fats, whereas the eleuroplast store proteins. Majority of the chloroplast of the green plants are found in the mesophyll cells of the leaves. These are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles having variable length, 5 to 10 micrometer, and width, 2 to 4 micrometer. Their number varies from 1 per cell of the Chlamydomonas, a green algae, to 20 to 40% in the mesophyll. Like mitochondria, the chloroplast are also double membrane bound. Of the two, the inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. A number of organized flattened membranous sacs called the thylakoids are present in the stroma. Thylakoids are arranged in sac-like piles of coins called the grana, or the intergranal thylakoids. In addition, there are flat membranous tubules called the stroma laminae connecting the thylakoids of the different grana. The membrane of the thylakoid encloses a space called a lumen. The stroma of the chloroplast contains enzymes required for the synthesis of carbohydrates and proteins. It also contains small double-stranded circular DNA molecules and ribosomes. Chlorophyll pigments are present in the thylakoids. The ribosome of the chloroplast are smaller, 70S, than the cytoplasmic ribosomes, which are of 80S. Ribosomes. Ribosomes are the granular structure first observed under the electron microscope as dense particles by George Palladay in 1953. They are composed of ribonucleic acid, which is RNA, and proteins, and are not surrounded by any membrane. The eukaryotic ribosomes are 80S while the prokaryotic ribosomes are 70S. Each ribosome has two subunits, larger and smaller subunits. The two subunits of 80S ribosomes are 60S and 40S, while that of 70S ribosomes are 50S and 30S. Here S, Sedberg's unit, stand for the sedimentation coefficient. It is indirectly a measure of density and size. Both 70S and 80S ribosomes are composed of two subunits. Cytoskeleton. An elaborate network of filamentous proteinaceous structures consisting of microtubules, microfilaments, and intermediate filaments present in the cytoplasm is collectively referred to as the cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. Cilia and flagella. Cilia and flagella are hair-like outgrowths of the cell membrane. Cilia are small structures which work like oars, causing the movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that of eukaryotic flagella. The electron microscopic study of a cilium or the flagellum show that they are covered with plasma membrane. Their core, called axonema, possesses a number of microtubules running parallel to the long axis. The axonema usually has 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of axonemal microtubules is referred to as 9 plus 2 array. The central tubules are connected by bridge and is also enclosed by a central sheath, which is connected to one of the tubules of each peripheral doublets by a radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Both the cilium and flagellum emerge from centriole-like structure called the basal bodies. Centrosomes and centrioles. Centrosome is an organelle usually containing two cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar materials. Both the centrioles in a centrosome lie perpendicular to each other, in which each has an organization like a cartwheel. They are made up of 9 evenly spaced peripheral fibrils or tubulin proteins. Each of the peripheral fibril is a triplet. The adjacent triplets are also linked. The central part of the proximal region of the centriole is also proteinaceous and called the hub, which is connected with the tubules of the peripheral triplets by radial spokes made of protein. The centrioles form the basal body of cilia or flagella and the spindle fibers that give rise to the spindle apparatus during the cell division in animal cells. Nucleus. Nucleus as a cell organelle was first described by Robert Brown as early as 1831. Later, the material of the nucleus stained by the basic dyes was given the name chromatin by Fleming. The interphase nucleus of a cell when it is not dividing, has highly extended and elaborate nucleoprotein fibers called chromatin, nuclear matrix, and one or more spiral bodies called nucleoli. Electron microscopy has revealed that the nuclear membrane, which consists of two parallel membranes with a space between 10 to 50 nanometer called the perinuclear space, forms a barrier between the material present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears ribosomes on it. At a number of places, the nuclear envelope is interrupted by minute pores, which are formed by the fusion of its two membranes. These nuclear pores are the passages through which movement of RNA and protein molecules takes place in both directions between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell. Variations in the number of nuclei are also frequently observed. Can you recollect names of the organisms that have more than one nucleus per cells? Some mature cells even lack nucleus. Example, erythrocytes of many mammals and sieve tube cells of vascular plants. Would you consider these cells as living? The cellular matrix or the nucleoplasm contains nucleolus and chromatin. The nucleoli are spherical structures present in the nucleoplasm. The content of the nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane-bound structure. It is a site for active ribosomal RNA synthesis. Larger and more numerous nucleoli are present in cells actively carrying out protein synthesis. You may recall that the interphase nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin, but during different stages of cell division, cells show structured chromosomes in place of the nucleolus. Chromatin contains DNA and some basic protein called histones, some non-histone proteins, and also RNA. A single human cell has approximately 2-meter long thread of DNA distributed among its 46 23 pairs chromosomes. You will study the detail of DNA packaging in the form of a chromosome in class 12. Every chromosome, visible only in dividing cells, essentially has a primary constriction or the centromere, on the sides of which disc-shaped structures called kinetochores are present. Centromere holds two chromatids of a chromosome. Based on the position of centromere, the chromosomes can be classified into 4 types. The metacentric chromosomes has middle centromere forming 2 equal arms of the centrosome. The submetacentric chromosome has centromere slightly away from the middle of the chromosome, resulting into 1 shorter arm and 1 longer arm. In case of arocentric chromosome, the centromere is situated close to its end, forming 1 extremely short and 1 very long arm, whereas the telocentric chromosome has a terminal centromere. Sometimes a few chromosomes have non-staining secondary constrictions at a constant location. This gives the appearance of a small fragment called the satellite. Microbodies. Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal cells. Thank you so much for listening to Neet Biology Readings. Please don't forget to like, share, comment, and subscribe.